Now, I will make an introduction to genome-scale metabolic models via an overview of the literature. Genome-scale metabolic networks link a sequenced annotated genome to metabolism. So for this, in a genome of an organism, all the genes that code for it are identified first. Okay, so enzyme coding genes are identified, and enzyme databases are used to identify the reactions that are catalyzed by those enzymes. So with this, you will have a link of gene, enzyme, and reaction. As an example, for example, in human genome, there is MDH1 gene, and this gene is coding for the enzyme known as malate dehydrogenase. This is the EC number for the enzyme if you go to any enzyme database such as Expazi or Brenda. And the corresponding reaction catalyzed by this reaction is the conversion of malate to oxaloacetate, a reaction in TCA cycle. So for each reaction in a genome-scale metabolic network, you will see the gene, enzyme, reaction, and pathway information. By using this genome, genes available in the genome, those with enzyme, those that code for enzymes, at the end, you will have the list of all possible reactions in that organism with their gene, enzyme, and pathway associations. This is referred as a draft genome scale metabolic network because you need to do some additional revisions to make it ready for simulations. By the way, genome scale metabolic networks is abbreviated in literature sometimes as GSMM, so the initial letters here, genome scale metabolic network models. M stands for model here. Some researchers, they refer to it as GEM, genome scale metabolic models, and some papers, they use this abbreviation genre. In my lectures, I will use GMN to refer to genome scale metabolic networks. In literature, for example, PPIN is used as an abbreviation for protein-protein interaction networks, and GRN is used to refer to gene regulatory networks. So to be on the same line, I use GMN to refer to genome scale metabolic networks. So all have those network parts at the end. This is a figure from a review paper published in 2009 that summarizes what I have just shared with you. The genome annotation, genes available in a genome, is used to identify the enzymes and the corresponding reactions. But also, there are several reactions that are known to take place in that organism, but somehow this information doesn't come from genome annotation. So you should also check publications to identify reactions that are known to take place in your organism. So this is the model building stage. And then with some other refinement steps at the end, you will have your genome scale metabolic models. The first genome scale metabolic network paper was published in 1999 in literature by Bernard Paulson and his PhD student Jeremy Edwards. So this is the first genome-scale metabolic network paper in literature, 21 years ago, published in Journal of Biochemistry, Biological Chemistry. And it was about Haemophilus influenza, a bacteria that causes flu. Haemophilus influenza is the first free-living organism with entire genome sequenced. Its genome was sequenced in 1995, and 4 years later, its genome-scale metabolic network was published and the related model was available. The model included about 500 reactions, about 350 metabolites, and 400 genes that control those reactions. I won't go into detail, but you can go and check. In the paper, they have used FBA, flux balance analysis, to simulate the phenotypic behaviors of Haemophilus influenza. They also performed gene deletion analysis. I will talk about this analysis approach in the coming week. So this was the first work available in literature. Later, the same group published in 2000 a genome-scale metabolic network of Escherichia coli. Compared to the model for Haemophilus influenza, this was a much more important breakthrough, because Escherichia coli is very commonly studied organism compared to Haemophilus influenza. Many researchers in the world, for several purposes, perform their scientific research is based on Escherichia coli. So availability of Escherichia coli genome-scale metabolic network was really a breakthrough in the field. So it is the second genome-scale metabolic network in science, and it is the second bacterial genome-scale metabolic network. It was published in PNAS, a highly reputable journal, and so far this paper was cited by more than 1,000 times. Those citation numbers are from Google Scholar, by the way. So compared to the first one, although it is the first one, it was cited by about 500 times, but E. coli, because it is, as I said, much more commonly studied microorganism, the work was cited almost twice more. The first Escherichia coli genome-scale metabolic network model included 720 reactions, about 400 metabolites, and 660 genes. And the initial naming for genome-scale metabolic models depended on this representation. Sorry, let me write it again. So this was used to refer to a genome-scale metabolic network. It was again suggested by, because they were the ones who reconstructed those genome-scale metabolic models. I here means it's an in silico representation of the organism. G and A is the initials of the developers. The PhD student had developed this work. And this is the number of genes in the organism. So this first model is referred as IGJE660. They have also applied in silico gene deletion simulations. So they have used FBA-based simulations to test the effect of deletion of 79 genes, if deletion of those genes lead to a lethal organism or not. And when they compared this with the literature, 68 of the deletions were in accordance with the literature-based experimental results. So in silico gene deletion simulations were successful with an accuracy of 86%. This again showed the power of genome-scale metabolic networks. For the first time, by including also gene information in the metabolic network, they have shown that you can simulate if deletion of a gene is lethal for the organism, for a microorganism, or not. This is a table from a paper published in 2003, again Paulson's group, 13 years of building constraint-based in silico models of Escherichia coli. So they have reviewed, in this review paper, available Escherichia coli models, and most of them actually they had developed. For example, this is a small-scale model with about 50 reactions. So Varma and Paulson published 3 or 4 papers by using this Escherichia coli model. And this was the earliest paper that used Escherichia coli within a mass balance-based framework. This is the Genomescale model, the first Genomescale model that I have mentioned, IGA660. And this is the updated version, IJR904. So they have updated this model 3 years later, and they increased the gene coverage from 660 to 900. Over the years, genome-scale metabolic network reconstructions were made available for many different organisms. Here I will review yeast and human genome-scale metabolic network reconstructions. The first genome-scale reconstruction of Saccharomyces cerevisiae, the yeast, was published in year 2003 in Genome Research. And this was a collaboration, the result of a collaboration between Paulson's group in UCSD and Nielsen's group, then in Technical University of Denmark. Jochen Förster was PhD student of Jens Nielsen, and Iman family was the student of Bernard Paulson. So two PhD students in two different research groups, they worked together to make available the first eukaryotic genome-scale metabolic network available. This was another breakthrough because the molecular details of Escherichia coli is much more simple. There is no compartments in Escherichia coli, no mitochondria, etc. So it was more challenging to come up with a metabolic network of a eukaryote. You have these compartments, mitochondria, nucleus, etc. So they say that the metabolic reactions were compartmentalized between cytosol and mitochondria, and transport steps between compartments and environment were included. They also applied gene deletion analysis, not here but in another paper. I will not go into details. So they published 3 or 4 papers as a result of this collaboration after they have reconstructed the model. This is from another paper, from a review paper indeed, by Nielsen's group, 15 years of large-scale metabolic modeling of yeast developments and impacts. So they have reviewed metabolic network models developed for yeast Saccharomyces cerevisiae. The first such model was published by Van Gulik and in year 1995, and it included 70 reactions. Then there was Nissen paper. We did a class exercise out of it, which included 37 internal reactions. By also including exchange reactions, the number of total reactions was 53. And of course, in those models, there wasn't any gene information. They had lumped reactions. They didn't have too much detail when it comes to biosynthetic reactions. And then the first model, IFF708, was published. So this model is referred as, by using the same convention, in silico model, FF. Since there were two students involved in developing this model, their surnames, initials were used, and the total number of genes was 708. So this first eukaryotic genome scale metabolic model is referred as IFF708 in literature. This is that model. And this was 2003. And the review paper is from 2012, 9 years later. So after this IFF708, many different versions, updated versions of Saccharomyces cerevisiae, the yeast genome scale metabolic network model, was published. And here you see how the number of genes, by using the green lines, you see how number of genes changed over time. The first model had 708 genes. So it increased to 800s and 900s and 930 over time. Also, number of included reactions increased too. So here, Yeast 4 has 1,800 reactions. Another thing, so far, the naming convention was I and the initials of the developer and the gene name. Here, later they started to name it Yeast 1, Yeast 4, Yeast 7, Yeast 8, etc. So the naming of the genome scale metabolic network models for yeast changed over time. Let's go through some of those papers mentioned in literature. So it's not here because this was 2012, but in 2013, Nielsen's group published a the most recent version then of yeast genome scale metabolic network, and it was named ITO977. So initials of the developer and the number of genes. This is a table from that paper which compares different yeast genome scale metabolic network models. Before talking about this table, let me draw your attention to one point. Since science on molecular biology produces many results, over the years, developments in measurement techniques, transcriptome, metabolome, proteome area, new functions for the genes are identified over time. So that's why you need to update the genome scale metabolic model over years. With the newly collected information in literature, you can increase, for example, there will be genes that were not associated with metabolism before, but with the new experiments, it is known that that specific gene is actually coding for an enzyme that catalyzes a reaction in trehalose metabolism, for example. So here they compare their model ITO977 with the other models in terms of the number of reactions, for example, number of cytosolic reactions number of mitochondrial reactions. So in the first model, there was 100 reactions in mitochondria, and IT0977 includes 250 reactions. Let's go on. This is a paper from 2010. They published Yeast 4.0. So I have wanted to share this because Pınar Pir, our faculty member, was also among the developers of Yeast 4.0. So this is from 2013. They said they have developed Yeast 6 of the Genomescale metabolic network, and they reported some improvements, especially based on gene essentiality, so these gene deletion simulations. They have shown that previous version, for example, had a false positive rate of 12%, and in Yeast 6, by refining the model, updating it, they have a lower false positive rate, and they have even slight increase in the true positive rate. So these are the things that you will see if you look at those update papers. They will report somehow that the new version of Genomescale metabolic network for that organism is better because of this and that reasons. So somehow a better prediction of growth rate, for example, or a better prediction of knockout gene deletion simulations. Now the most updated, the most current version of Yeast Genomescale metabolic network is maintained by the systems biology group in Chalmers, by Jens Nissen's group, and you can use this GitHub page to access this version, and it is referred as Yeast Gem, and the current version is 8.4.2, so version 8, and it includes about 1,100 genes, 4,000 reactions, and the related paper was published in 2019, a consensus Saccharomyces cerevisiae model Yeast 8 and its ecosystem for comprehensively probing cellular metabolism. So this is the most recent version of Yeast Genomescale metabolic network, and they continue to update it. If you are interested in, you can check the GitHub page. There is one interesting point, however. In this paper published in 2016, is also a co-author, and the leading author is Isabel Rocha from Portugal. The paper says, improving the flux distributions simulated with genome-scale metabolic models of Saccharomyces cerevisiae. And please look at the first sentence in this paragraph that I have copied here. In their conclusion section, they say that we observed that the oldest genome-scale metabolic model of Saccharomyces cerevisiae, IFF708, showed the best prediction of central carbon fluxes. Yeah, this is really surprising, right? They also share the prediction graphs here. So they have used measured central carbon metabolic fluxes. They compared it with the predicted fluxes when using different yeast genome scale metabolic models. So IFF708, for example, had a prediction R-square of 0.88. ITO977, published in 2013, 10 years later than the first one, had an R-square of 0.82. IMM901, another model, had an R-square of 0.76. 0.78, and yeast 6 had an R-square of 0.56 because of this prediction. So interestingly, the first model had the best prediction. So my point is, yes, they have improved the models in terms of gene coverage. This is important. Of course, there are more genes available here, more reactions, so you have a better coverage of the genome of the organism, also the metabolism of the organism, because of additional reactions and additional pathways, additional genes. And maybe in terms of gene deletion simulations, you simulate many more conditions with a higher accuracy with the new models. But in terms of predicting fluxes, the new models did not do a good job. So it doesn't mean that a new model is perfect or much better in all different aspects. That's not the case. I will also talk about Genomescale networks of human metabolism. The Genomescale model of human was published in 2007, and it was named as Recon 1. Later, newer version of this model was available, and the most recent version is Human Gem. I will talk about those. So this is the first paper published in 2007. Again, Bernd Paulsen was the leading scientist in reconstructing human genome scale metabolic network, and it included about 1,500 genes in human genome, and the number of reactions you see here. And one interesting thing, there were so many compartments included in this model. So here you see their names, and here they say that full compartmentalization of metabolites in cytoplasm, mitochondria, nucleus, endoplasmic reticulum, Golgi apparatus, lysosome and peroxisome, and extracellular environment were considered. So the thing is, they have also separately considered if an enzymatic reaction takes place in endoplasmic reticulum, in lysosome, etc., in addition to cytoplasm and mitochondria. Later, they have published the second version, Recon 2, 6 years later in 2013. Again, the leading author is Bernard Paulsen. You see many more other scientists well known in the area, such as Hans Westerhoff from Holland, Douglas Kell from UK, Pedro Mendes, I think at the time he was in UK, and also Jens Nielsen was among the developers. And here, a table from that paper that compares Recon 1 and Recon 2. Number of unique genes increased from 1,500 to 1,800. Number of reactions increased from about 4,000 to 7,000. Number of metabolites also increased from about 3,000 to 5,000. You also see specific number of metabolites in each different compartment, different organelle. They also gave the size of their stoichiometric matrix, their number of linearly independent mass balances, etc. This is a table from a book chapter I co-authored. And here you see a comparison of Recon 1, Recon 2, and other stoichiometric genome scale metabolic networks from literature. Later in 2015, HMR2 Human Metabolic Reactions Metabolic Network was published by Nielsen's group. Compared to Recon 1 and Recon 2, it included much higher number of genes and higher number of reactions and higher number of pathway associations. My point is, these are all generic metabolic networks. If you consider human metabolism, the metabolism has differences in brain or in liver or in kidney. There are reactions that are active in kidney but not active in brain. There are pathways that are active in liver but not active in brain, for example. So in these generic metabolic networks, all possible reactions were included without making any distinction between for the organs, for the tissues. But if you want to specifically analyze a specific organ by using metabolic network approach, you will need organ-specific or tissue-specific genome-scale metabolic networks. We have indeed developed a brain-specific genome-scale metabolic network in year 2014. It included 570 genes, higher than a previous brain-specific metabolic network. And so it was named IMS 570. It was developed by Mustafa Sertbaş, so the initials MS you see there. Later again, Paulson's group published in 2018 Recon 3D. And they also have a website, Virtual Metabolic Human, associated with Recon 3D. You can go and you can search the reactions. These are some statistics from Recon 3D in terms of number of genes, number of reactions, number of metabolites in comparison to other genome scale metabolic human metabolic networks in literature. So in Recon 3D there are 13,000 reactions, 3,000 genes. It also includes information on the variants on the genes and also the structure linked to the structures, protein structures available in the annotation information. So the most current, the latest version of human genome scale metabolic network was published in 2020 by Nielsen's group. You can access the GitHub page by using this link. This is the most current version 1.5. And here you see the number of reactions and number of genes. It includes 3,600 genes and about 13,000 reactions. In reconstructing human GAM, this most recent version, they have used Recon 3D, they used HMR2, their previous model. They also used tissue-specific models such as iHepatocyte, which is a genome scale metabolic model for liver cells. There is an associated website, metabolicatlas.org, very useful. If you go there, you can search for the reactions available in human GAM. You can search for the reactions, for the genes, for the pathways, and it will give you the corresponding lists. It also includes associated with the related pathways in human GAM.